Hi, my name is Alessandra Madigan, and today I will be talking about the threats PCBs and microplastics pose on killer whales and baleen whales. I will start by talking about the natural cycle of nutrient runoff, and then I will talk about the anthropogenic impacts on this cycle, such as eutrophication and PCBs. Then I will talk about bioamplification, bioaccumulation, lipid impact in blubber, microplastics. And then in my discussion, I will compare and contrast between my two study cases, which are killer whales with PCBs and microplastics with baleen whales. Finally, I will conclude this presentation with the major takeaways. Nutrient runoff is a naturally occurring process that is disrupted by the over-enrichment of practices such as agriculture. The stages of nutrient runoff include coastal upwelling, tidal mixing, runoff from land, and overflow from rivers. The nutrient enrichment cycle is important because it provides excess nutrients to marine ecosystems, which is essential for the growth of aquatic plants. And in terms of the anthropogenic impacts, when there is an exacerbation of pesticides like PCBs, it can be harmful to the levels of nutrients found in marine ecosystems and freshwater ecosystems, which can cause eutrophication, and that is harmful to aquatic plants and other species, such as killer whales and other cetaceans. Lipids and the influence in blubber. Blubber is important for cetaceans, such as killer whales, because it is a primary factor enabling multiple processes in the body. For example, when PCBs affect species like killer whales, blubber is the main absorber of bioaccumulation of these levels of PCBs. And this is because blubber is the primary site for lipid storage. During lipid mobilization, for example, starving and lactation, in killer whales, PCBs are released from the blubber into the bloodstream. This can cause high transfer exposure to young calves through milk. Now to talk about lipid storage. Lipid storage primary site is within the blubber. Lipids are activated when animals are in distress as a mobilization process within the body. My case study for PCB contamination focused on killer whales, so orcas. The study focused on North and South residents and found that significantly higher levels of PCBs were found in southern residences over northern residences, which meant that when they used modeling to detect future projections, which will be spoken about more later, they found that southern residences were more likely to decline in population more rapidly over northern residences. A very important take from this study is that killer whales can live up to 90 years, which means that bioaccumulation is accumulating over all of that time. And something else that was found within this study was that adult males faced the most health threats the older they got. So now we're going to break down eutrophication, PCBs, and bioaccumulation. So eutrophication, as seen in the photo in this slide, occurs due to an exacerbation of nutrients in fresh and marine ecosystems, as well as the addition of PCBs and other pesticides from anthropogenic use. Eutrophication impedes on the natural process of nutrient runoff, and it also can cause algal blooms. PCBs also impede on the natural process of nutrient runoff, And as spoken about in the last slide, it enters through the blubber, and then through lipophilic bioaccumulation and lipid mobilization, it enters the circulatory system, which impacts mainly orca calves through milk from their mother and adult males through future bioaccumulation as they get older, as spoken about in the previous slide. PCBs found in cetaceans such as killer whales can affect the liver and thyroid hormone development in calves through drinking their mother's milk and also consuming it at a young age directly. And this is because killer whales and other cetaceans lack a specific enzyme known as the cytochrome P450 enzyme, which allows for filtration in species that have this enzyme of insecticides such as PCBs from the liver, gut, and lungs. And since killer whales and other cetaceans live such long lifespans, it will take them a really long time to catch up with the effects of anthropogenic climate change, such as contaminations like PCBs. Now to move on to my second contamination, which is microplastics. As you can see from the figure to your left, microplastics come from many everyday consumer goods, such as clothing and beauty products, and can take thousands of years to fully decompose. Microplastics can be broken down into two different types: microplastics and microfibers. Microplastics range in size from 0.001 to 5 millimeters, and microfibers range in size from 0.8 to 0.9 millimeters. And the average diameter of microplastics are 16.7 mu. So just some statistics about microplastics. 83 million metric tons of newly produced microplastics still exist on our planet today. And since 2015, 6,300 metric tons can be found in landfills, marine ecosystems, and terrestrial environments. Microplastics can impact marine environments either directly through consumption or indirectly through the trophic levels and bioremediation. In baleen whales, which is the focus of the case study with microplastics, they're impacted both directly and indirectly. They're impacted directly through inhalation or consumption through the baleen, or indirectly through consumption of contaminated prey, such as krill, that have also eaten microplastics themselves. Baleen also feed at a specific depth between 50 and 250 meters in the pelagic ecosystem. This is a depth known for the highest contamination of microplastics to exist in the ocean. Now to move on to the discussion. So these are the challenges and impacts in both case studies. So you can say that in both studies, these species were impacted both directly and indirectly by these contaminations, but the PCBs in killer whales was definitely more of a direct. This was through either dietary consumption or bioaccumulation over the duration of their lifetime. As spoken about in previous slides, bioaccumulation and the consumption of PCBs impacted hormone disruption and thyroid and other growth factors in calves. Methods used in the PCB study was mainly modeling and biopsies on deceased orcas. The same can be said for research methods found in the baleen whale study for microplastics, except they also had the ability to tag baleen whales and perform more biopsies. which allowed for more in-depth information and outcome to come out of the microplastic studies with baleen whales over PCB contamination study with killer whales. Even though this was to be the case, there's still room for a lot more research to be done, as there's a lot of areas that are lacking in in-depth knowledge and understanding. So some major takeaways. PCBs are chronically affecting the chemical and biological development of reproduction and offspring of killer whales. Due to lack of research and small sample sizes, it's hard to tell what the long-term effect will be on populations such as the North and South residences, especially as increased amounts of PCBs and other pesticides enter marine ecosystems due to increased overconsumption. Microplastics pose a physical health threat on baleen whales in terms of foraging patterns, and overall these case studies showcase that these contaminations pose a threat to all levels of marine ecosystems, especially top marine predators such as orcas and baleens. Thank you.